In this video, we'll go ahead and quickly finish out the last of the two steps in animal virus replication. After you have your synthesis step, you'll have assembly and maturation. In assembly, the subunits or the components of the new variants that were produced during synthesis come together and assemble. In many cases, this is a self-assembly process. In other words, there's no enzymatic function or activity associated with it. You can see also that this would ultimately allow for the viral genome to be placed within the capsid as well. The site of assembly does vary with the type of virus, and we'll talk a little bit about how that ultimately will affect its release. The release is when the virus is going to leave a cell. And the release of the virus really depends upon whether or not that virus has an envelope or does not. Enveloped viruses actually have more options for release than naked viruses. Naked viruses always exit the host cell via exocytosis, typically inducing apoptosis or destruction of the cell as they leave. However, if we begin to take a look now at the enveloped viruses, they typically exit the cell via a process called budding. With budding, the enveloped virus is essentially going to take a piece of the host cell's membrane with it. As you can see here, the nucleocapsid approaches the host cell's cytoplasmic membrane. Ultimately, in that membrane, as the virus continues its replication process, viral proteins that ultimately will become spikes are incorporated. The spikes can then bind the nucleocapsid. The nucleocapsid then begins to proceed to push, for lack of a better way of putting it, push its way out toward the external area of the cell. Ultimately, as it continues to leave, the cell evaginates. In other words, it extends its cell membrane out, and the cell membrane will begin to wrap around and coat or cover that particular nucleocapsid. As it does so, it eventually will pinch off, and once it does, notice the host cell's membrane is left intact, and what is left is a piece of that membrane containing the spikes surrounding the previously naked virus, and at this point it would become an enveloped virus. So the enveloped virus is released via budding, essentially a process where they will take a part of that host cell's membrane with them as they leave. Key thing keeping in mind, the host cell is not damaged or killed here. Well, it's damaged, but it's not going to be killed as a result of this process. These non-enveloped viruses typically don't do that. They usually induce apoptosis upon their release. Apoptosis is actually programmed cell death, so the cell is destroyed or killed in the process of releasing the virion. I want to briefly now mention acute versus persistent infections. The most simple of these, or straightforward I should say, would be an acute infection. Acute infections have typically very rapid onsets and short durations. Essentially, you get infected, you show signs and symptoms of the disease, your immune system hopefully fights back, and ultimately, once it's resolved, you show no signs of infection. Let's take a look at that here in this top area. Notice that over the course of a few days, this person may have been infected. That just means the virion is present. They may not know it yet, but it's there. As the virion infects more and more host cells, that person transitions to a point of disease. This is when they would show signs and symptoms. You see they use the example of influenza. After a period of hopefully a matter of days, usually 10 to 14 days, your immune system begins to fight back adequately enough in order for the amount of virion to begin to decline. But during this stage, you would also still be infectious because the virion would be present. Once your immune system completely resolves the infection, you can see the infectious virion number should come down to 0. That's an acute infection. I usually like to say it as you get sick, for a short period of time, you get better, and then the virus is gone. We compare that with persistent infections. Persistent infections, well, they persist, meaning they last long periods of time. Persistent infection as it says here in the slide, can last years or even your whole life. And at times you may have symptoms, and at other times you may not. Examples for some viruses that exhibit both persistent and acute infections. Well, major example would be HIV. It can cause an acute infection, especially upon its first introduction. A lot of symptoms resemble the flu, for instance. So you're sick, you have virion, and you experience those symptoms. You'll get better, hopefully, but then the virus would still be present. So it would sit back and last for a long period of time. So it can be both an acute and a persistent infection. Persistent infections come in two different varieties. You'll have your chronic infections, in which the amount of virion being produced would be consistent over a long period of time. In other words, you're actively shedding or producing virus over a long period of time. And then you have your latent infections. Latent infections come and go. In other words, you have periods where you're actively producing virion and you can detect it. And then, well, your body resolves it and the virus essentially goes dormant for a short period, for however long it may be. And then ultimately, it can reactivate. And when it reactivates, you would have another productive infection. Let's take a look at that very quickly here with the bottom two images. This image details for you the chronic infection of hepatitis B. Notice the virion is introduced, the virion numbers increase, but you wouldn't have active or rather show signs and symptoms of disease yet. You show your signs and symptoms of disease, your immune system fights back, it begins to resolve those signs and symptoms. However, look at the amount of virion. It never declines. You can see in time, they say it persists for years. This is an example of a chronic infection. You get sick, you have signs and symptoms. Yes, those signs and symptoms go away, but you continue to produce virus at a level that can be detected for a long period of time. Contrast that with the latent infection seen by the herpes simplex virus 1, or the virus that causes cold sores. You can have an active infection in which you'll have the cold sore around the oral cavity. The immune system resolves it, and then in between, there are no virions being produced, so that person would not be infectious. At a later point, that virus would reactivate, and you would get the signs and symptoms of the disease all over again. Notice here, we're not having this chronic sustained production of virion. Note, instead, the production of virion goes to zero, you're non-infectious, and then, well, it recurs later on once reactivation happens. Multiple viruses are usually going to exhibit the example of being a persistent infection. You can see viruses such as the cytomegalovirus, which is known to cause some congenital birth defects. That's one of the major concerns that we usually look at that with. EBV, or Epstein-Barr virus, causes ultimately causes mononucleosis, but can also cause some forms of cancer such as a Burkitt's lymphoma and some other carcinomas. The Hep B and Hep C viruses, both of these are chronic viruses, and they ultimately can cause hepatitis, but long term, they can lead to liver cancer. So you see hepatocellular carcinoma. That's liver cancer. I mentioned to you a moment ago the herpes simplex virus type 1, HSV1. That's a latent virus which causes cold sores. HSV2, also a latent virus, That latent virus is going to be the culprit behind genital herpes. HIV actually exhibits mixed. You can see the chronic, it actually affects the active helper T cells. Latent, it affects the memory T cells. We'll talk about those actually in our immunity chapter. Then the varicella zoster virus, which actually causes chickenpox. It's a latent virus. If you think about it, shingles, which is the latent form of it, can be reactivated multiple times over the course of an individual's life. To finish this chapter out, we want to quickly mention that some human viruses are associated with cancers or tumors. As it says here, cancer or malignant tumor is one that can move. It can go from one area to the body to another. Benign tumors don't move, and they would be considered non-cancerous. Within the cell, you have what are called proto-oncogenes. These can stimulate cell growth and division. And then you also have, in contrast to those, tumor suppressor genes, which inhibit growth. If for some reason a virus enters a cell and causes mutations in either of those gene pools or gene components, you can ultimately see a tumor or a cancerous condition arise. An oncogene is a proto-oncogene that has ultimately been transformed in order to allow it to promote uncontrolled cell growth. Multiple viruses are associated with a variety of human cancers. But the key to understanding what's happening, or what I'm trying to say here, is this. The genes required for abnormal growth are always there. Those are your proto-oncogenes, and the ones that stop them, the tumor suppressor genes, are always present. Mutations in either could lead to uncontrolled cellular growth, which is a cancer. There are a multitude of viruses typically associated with various human cancers. You heard me mention in the previous set Epstein-Barr virus. Told you it causes mononucleosis, but it also causes Burkitt's lymphoma, which is a B-cell type of lymphoma typically seen affecting African males, nasopharyngeal carcinoma, which usually affects Asian males, and then the B-cell lymphoma is just kind of the generalized cancer there. Both of these typically are B-cell lymphomas. This is just generic. Hepatitis B and C, you can see here and here, cause hepatocellular carcinoma or liver cancer. Herpes virus type 8, usually seen more so in people suffering from HIV or AIDS. It causes a skin cancer called Kaposi sarcoma. HPV, associated with a usually pretty well known because it causes cervical cancer in women. And then you have HTVL1, an RNA retrovirus that actually causes adult T-cell leukemia, and that is extremely rare, as it says here in the slide.